group of cousins that you don't want. They're really, they're set apart from some of the others because of the level of disease they produce.
So where do you stand on this question of whether viruses are alive or not?
Well, viruses are not living things, in my view. They're totally dependent on the cell, and a living cell. They have to get into the living cell and then use the living cell's machinery to make more of itself. So we don't typically consider them living things, but they cause a lot of problems for the living.
So then what makes the coronavirus family special?
Well, it's a very big family, for one thing. There are a lot of different coronaviruses in nature, and a lot of the animals, and they can cause disease in them. In humans, it circulates all the time, these coronaviruses. It's called endemic. It means it's circulating in people all the time. But typically, these viruses cause a mild respiratory disease like a cold. So they're not considered a major problem. But they are responsible for 15 to 30% of all respiratory illnesses in people each year. But not like it is right now. There are 4 different groups of coronaviruses: alpha, beta, gamma, delta. The alpha and the beta are the ones that cause disease in mammals. And the current COVID virus is a member of the beta coronaviruses. And that includes SARS and MERS. This is a nasty group of cousins that you don't want. They're really, they're set apart from some of the others because of the level of disease they produce.
What are the, structural characteristics or the replication characteristics of the distinct types of coronaviruses that make some of them pathogenic and others not, and some of them extremely dangerous, like this one, and even more so, SARS and MERS, and others a common cold?
Well, we don't have all the answers to that.
Sure.
Clearly, pathogenicity is a huge issue. But for one thing, the receptors they recognize, like in the case of this coronavirus, it recognizes a receptor that's quite common in the lung. And so therefore it's able to infect and replicate there probably more efficiently than some other viruses. It also can infect macrophages and dendritic cells. And what that does is it doesn't produce more virus in those particular cells, but it causes an explosion of cytokine production by the host, by us. And that can lead to a cytokine storm, which also then damages the lungs. So this one, there are a variety of factors: how much virus they produce, how quickly they spread, all those types of things make a difference too. But this virus does have some unique characteristics with regard to those 2 factors.
Are most of the viruses that have caused us so much trouble recently zoogenic viruses, that they've come from animals? Is that?
Zoonotic. Oh, yes. Well, the SARS and MERS clearly were thought to be, because this group of coronaviruses is thought to originate from bats. And they have a large number of coronaviruses, bats do. Doesn't seem to bother them much, but they're the major reservoir, apparently. And these 3 viruses certainly have caused more severe disease and death than any of the others that we've seen. Certainly a lot more than the ones that just cause common colds.
And are there reasons known why bats coexist with them so peaceably and we do not?
Well, there's a lot of work being done on that, and there are a lot of theories at this point. And so they think maybe that bats are the natural reservoir and the viruses have evolved with them. And also the bats, when they fly, which they do, they build up their metabolism, and metabolism speeds up, which may help their immune response be better. And also that they develop a high body temperature, like our having a fever. And so viruses don't like heat. And so they think that maybe those factors help it control these viruses so that they aren't a problem for them.
What makes, from what we, obviously the research is fast moving, makes this coronavirus distinct structurally from, or genomically from, the MERS and the previous SARS epidemics?
We don't really know. Maybe it picked up a mutation or some other genetic material that helped it become more transmissible. This one is highly transmissible, which the other 2, they didn't go spread easily in people. And so therefore they kind of died out. They did not move person to person easily.
Uh-huh.
And that's what it really takes for a virus to cause a pandemic.
I know that thousands of scientists around the world are working now on this particular virus. What are some of the most anticipated findings?
Well, certainly a lot of effort's going into producing a vaccine, but that takes a year at least to get out to the general population. So that's for potentially a next round, or if it continues to cause problems. Drugs that may be inhibiting the virus replication, those are certainly being tested, a lot of different ones. Also, I'm very much into the antibody testing, because if people recover and become immune, then they might well be able to go back to work. And so
Sure.
antibody tests to see if people have antibodies to the virus and therefore might be protected are also very important.
What are the kind of distinct features of the virus or the body's response to it that can provide us pathways toward vaccine development for this particular?
Well, this virus has some vulnerabilities because of its replication. And that's what they focus on a lot of times, is what steps in the replication could you either use a drug to interfere with that so that it doesn't produce as much virus or it controls it. So that's what you look at, first of all, is the replication process, because there's steps in there that make the virus vulnerable, because it'll block something that the virus needs. Like the virus uses cell machinery, but it also produces proteins and entities unto itself. And so if those are you're able to interfere with those, you can stop the virus. They're also looking if there's anything they can do to control the cytokine storm. Yeah, that's actually our body responding and over-responding.
Right. What makes antiviral drugs so much more difficult, at least from the outside it seems, to develop antibacterial drugs?
No, it's true. And the reason being is because the virus is using the cell's machinery. So it's very easy to interfere with the cell as well as the virus. And so you're trying to minimize the impact on the cell, the normal cell that's infected, and trying to not, and at the same time, suppress the virus replication. So it's a much more complex arrangement than it is with bacteria. Because viruses have to get into the cell and take over. And if you can block any of the steps where they've taken over and not damage cells, other normal cells at the same time, it's trickier because they're using the cell's machinery.
One tool that we have this time around that we didn't even as recently as the MERS and H1N1 is the ability to kind of rapidly sequence the genome of the different iterations of it. What can we learn from that, or how is that transforming our understanding and our response?
Well, certainly they look for relatives, and SARS and MERS are relatives, they're cousins of this virus. They look for additional genetic information that the virus has that maybe is responsible for making it so highly transmissible. And so that's one of the things they look for, is does that genome segment, if there's an additional segment in there, what does it do? And is there something special about it that we need to understand so that we can potentially block that?
You were about to explain a moment ago why the development of a vaccine takes quite some time, but I think that I should let you elaborate on that because, of course, people around the world are wondering why. As I understand it, 18 months would be miraculous.
Yeah.
Which is sort of what the number that people keep throwing around. But, what are the steps involved in challenges from conceptualization to shipment that make this such a daunting challenge to do quickly?
Well, I think, it is a long process, and it's mainly for the safety and to determine the efficacy. You start with a small group, then you move up to a large to see if it's safe. Then you move up to a larger group, then a larger group. So you go 10, 100,000, and then thousands. And then they can say, okay, it's proven to be safe and to protect people. Then you have to manufacture it and get enough doses for millions of people. So it's a lengthy process, but that process takes a good while just because of the number of people involved to test it.
Is it known clinically, what are the characteristics of this virus that are generating in some patients, such a kind of violent immune and debilitating immune response, and what isn't?
But they're certainly trying to figure that out. It is not clear. Is it genetically? different among folks so that they have a different response? Is the receptor different so that it either attaches more or less? There are a lot of possibilities out there as to why it's causing such a problem in people. And it's the old and the young. Now, the older people, particularly the kapuna, have a higher death rate because we have more comorbidities or more diseases or conditions that might impact on our health as well. For example, if you have pneumonia and your heart is having to work really hard because you're not getting enough oxygen, then you run into the problem of maybe your heart having a problem as well as your lungs. So that's the reason it's a higher death rate in older people, but it certainly, unfortunately, can kill young people as well. And sometimes they think that's a cytokine storm. In the 1918 influenza pandemic, many young people died within 3 days, and they actually thought that that might have been due to a cytokine storm because younger people have a more robust immune system than older people.
As you assess all of this research going on in different fields, or are you most optimistic about vaccine development or drug development or some other way of containing or treating this pandemic?
Hopefully drugs could get to market quicker, particularly if it's one that's being used for something else. It's also, I do think it's really important to really know the breadth of the disease because we were very late getting started on testing for this. So we didn't really have a good idea as to how many people were actually infected. Many people could have been infected and they didn't show any symptoms. So we need to get a better understanding of how many people have had it, and maybe they could go back to work, which would help a lot of people. So I would think those are going to be the things we need to do. But right now, we're still left with the more draconian measures like the physical distancing, quarantines, no travel, strict hygiene. That's all. That's what we need to do right now. And washing your hands. Your hands are really very, very important in this one because we use them. We touch a lot of things, and we touch our nose, our eyes, and our mouth unknowingly a lot of times. And so it's really important to use warm, soapy water. Warm because viruses don't like heat, that it can inactivate the virus. We use soap because this particular virus has a little fat coat around it, and soap dissolves fat. And then run lots of water over your hands because that'll dilute anything that's on your hands.
Okay, thank you very much.
You're more than welcome. Mahalo.
Okay, mahalo.